Chapter 26 is about nutrition and metabolism. Before we start, let me tell you some key points from Chapter 25 that I possibly missed in last presentation and that you need to know. So, the first key point is that the liver, it's an organ that it is in charge of making albumin, produce bile, and also is in charge of metabolic control and hematologic control. Another key point is that the pancreas releases insulin under the influence of gastroinhibitory peptide, or GIP. Now the key point is that the pancreas secretes a fluid that it is reaching bicarbonate ions through the action of secretin hormone, secretin just like secret. And then the pancreas secretes this pancreatic juice, that it's a fluid that has enzymes that can digest carbohydrates, proteins, and lipids. And one of these enzymes is trypsin. Trypsin, it's an enzyme that digests proteins into peptides. So those are the last key points for chapter 25. Now the key points for chapter 26, I will tell them in the middle of this chapter 26 presentation. And also, before we start, let me tell you something about the PowerPoint for the lab report. So in this PowerPoint, you have seen some of these models with arrows pointing at structures with numbers. So these are structures that you need to know their name at least, and then you don't have to provide me with the list of the names. So you need to know the names of these structures because they might be coming in the practical. Okay, now let's move into the chapter 26 material. So in order for us to make tissues and for these tissues to function, we have to have a source of energy that provides this material so you can perform all this biological work. And then this source of materials is also used for replacing worn out biomolecules or cells. And then the metabolism is defined as the chemical change that lies at the foundation of form and function. And we have within metabolism all these chemical reactions in our body that are subdivided into anabolism if you're making large macromolecules like DNA, proteins, out of the small raw materials like amino acids, etc. And then we have catabolism, which is the opposite of anabolism, in which you break down these macromolecules in order to form small molecules that can be utilized in other ways. And both anabolism and catabolism forms metabolism. Once we start ingesting nutrients, and then we have all these macromolecules form, we tend to store some of them. And then we can gain weight if the intake exceeds the output, and we can lose weight if the output exceeds the intake. Now, in general, our weight seems to be stable in a homeostatic set point. However, the weight can vary from person to person according to many factors, including heredity, environmental influences, according to your age, your physiological condition, etc. And then in general, 30 to 50% of the variation in the human weight is based on genes, inheritance. Okay. Now let's start with this first key point. which is the carbohydrate metabolism. So carbohydrates are very easily absorbed and used as energy from our body. And then uh, we use these different types of carbohydrates that at some point all end up in glucose. And glucose, it's a molecule that helps to form ATP. Now, this is the chemical reaction in here for glucose catabolism through oxidative catabolism. So we have C6H12O6 plus 6 molecules of water that are going to be converted into these byproducts: ATP, 6 carbon dioxide, and 6 molecules of water. Glucose is catabolized through a series of small steps, and these are controlled by different enzymes. And the actual glucose metabolism, catabolism is called glycolysis, and we have this glycolysis happening within the cytoplasm. And the ultimate byproduct of glycolysis, which is pyruvate, is going to enter into the mitochondria so you can generate more ATP. Now, as we are producing ATP, we also generate heat in the cells, and that heat is going to be released from the cells. And that helps to maintain a specific temperature so that the enzymes that are responsible for the metabolism can work at the appropriate temperature. So we have these major pathways for glucose catabolism. So the first one will be glycolysis. And in this case, you will form out of glucose, as I told you before, pyruvate. And glucose is a 6 carbon molecule that is going to be split through this series of reactions into this last metabolite of glycolysis, which is pyruvate, which is going to have three carbons. Now, as you are moving these glucose molecules and transforming into pyruvate, you will generate a couple of molecules that carry electrons. And then these electrons are going to feed into the mitochondria, and then you will generate ATP out of them. Now, anaerobic fermentation, it's the glucose catabolism under anaerobic conditions in which you don't have oxygen. And in this case, you will reduce pyruvate into lactate. And then in aerobic respiration, in which you use oxygen, you will oxidize pyruvate into CO2 and water. So this is the steps of glycolysis and anaerobic fermentation. We will see this in a greater magnification. That's the best that I could do with this figure. So this is the starting point for glycolysis. So you have these 6 carbon molecule represented by these 6 gray circles in here. This is glucose. So once glucose enters into our cell, glucose is going to be phosphorylated. So for this, you will use adenosine triphosphate, and then you attach one phosphate into glucose at the 6 carbon to form glucose 6-phosphate. And then this ATP, since it's losing one phosphate that you attach into these glucose molecules, is going to be converted into ADP. Now, when you do this, you will make this glucose molecule a little bit bulkier. And also, you will add a negative group from phosphate. What this is going to help for is that the glucose won't escape out of the cell where it's not needed. Now, this glucose 6-phosphate can proceed into 3 different pathways. It can go directly going down to form these pyruvate molecules, or it can go and be stored as glycogen through a different pathway, or go and be stored as fat into a different pathway. Now, before glucose is converted into fat, of course, it has to be utilized if you have certain metabolic needs. Whatever you have left out of this glucose after you have meet your metabolic needs is going to be first stored as glycogen in the liver and in the muscle cells, and then the rest is going to be stored as fat. Now, this glucose molecule, glucose 6-phosphate, then is going to be converted into an isomer, which is going to be called fructose 6-phosphate. And then this fructose 6-phosphate is going to be phosphorylated, and you will have now fructose 1,6-diphosphate. So you had fructose in carbon 6, and then you attach another one in carbon 1. That's why you call it fructose 1,6-diphosphate. And then you use your second ATP in glycolysis. And then you have ADP again. So up to here, up to these steps, you have used 2 ATPs. Now, this fructose 1,6-diphosphate is going to be split into 3 carbon molecules that we call 2-phosphoglyceraldehyde. And then these are going to be phosphorylated or oxidated, and then you will dephosphorylate these 3 carbon molecules, and then you will obtain your 2 ATPs in here. So as you are doing this dephosphorylation, you will add phosphate group from this part in here, from this molecule in here. You will add it into ADP, so you will have 2 ADPs coming, and then you end up with 2 ATPs. So up to here, you have no gain, no losses of ATP. Now in the second step of this dephosphorylation step, you will have again 2 more ADPs, and then you will remove to these molecules the 2 phosphate groups, and you add them to these 2 ADPs, and then you will have 2 ATPs, so you will have gained 2 ATPs. As you are going through these steps, you will also generate NADH. So you will have this nicotine adenine dinucleotide, which is a carrier of electrons. You will remove hydrogens from these molecules in here to phosphate phosphoglyceraldehyde, and then you will add these hydrogen ions into these 2 NADH, so you have 2 NADH plus 2 hydrogens. These will come through anaerobic fermentation and be recycled again, and then you can form 2 lactates. Now, if you go into this step, pyruvate is going to go into the mitochondria, so you can form more ATP out of here. If you just stop glycolysis at this step and you don't enter this pyruvate into the mitochondria, you will just make 2 ATPs, and this is not enough for producing enough energy. So in glucose catabolism or in glycolysis, you have these enzymes that helps to remove electrons as hydrogen ions, and then you will transfer them into this NADH through oxidation reduction steps. And then when NADH, that it has this plus sign, accepts hydrogen ions, is going to be converted into NADH. So, sorry, NAD. When NAD plus accepts hydrogens, it's going to be converted into NADH, and this is going to be the reduced form. So NAD is an oxidized form, and then when accepts these electrons from hydrogen, when NAD accepts these electrons, it's going to be reduced into NADH. Now we have then these hydrogen ions carried by NAD, and this NADH is going to transfer these hydrogen ions to coenzymes. And then these coenzymes are going to bring these hydrogen ions into other compounds that are going to ultimately generate more ATP. Now, in general, these enzymes in glucose metabolism cannot work without these coenzymes. So here are the two coenzymes then. NAD plus, which is called nicotinamide adenine dinucleotide. It is a derivative from niacin, which is vitamin B. And then NAD, as I told you before, is going to be in an oxidized form. When it receives the electrons from these two hydrogen ions, it's going to be reduced into NADH plus H plus. And then you can have the reverse of this reaction. So NADH, which is oxidized, when it donates the hydrogen ions into the other compounds of the energy transfer, it will become NAD, and this is going to be oxidized. So this one was reduced because it had these hydrogens coming from these electrons coming from these hydrogens, and then it's going to become reduced. And then when it releases these hydrogens, it's going to become oxidized. So this is the oxidized form, and this is the reduced form, and then they can cycle. And then we have a similar coenzyme, which is called flavin adenine dinucleotide, or FAD, which basically will do the same. It will be in oxidized form, and then when it accepts these electrons coming from hydrogen, it's going to become reduced, which is going to be now FADH2, and then it can go back into oxidized state. So hydrogens will be removed in their protons or their electrons in pairs, and then these are going to be transferred into coenzymes. And these coenzymes, as I said before, they're going to be carriers for the energy that is coming from glucose, so you can form more ATP. So the enzyme that phosphorylates glucose in glycolysis is known as hexokinase, and this one is going to transfer inorganic phosphate from the cytoplasm into the glucose, so you can have glucose 6-phosphate. And then, as I told you before, this is going to make a bulkier molecule that is going to be incapable of leaving the cell, and also you will have this negative charge coming from the phosphate group, preventing from as well the glucose exiting the cell because you have a negative charge. Now, glucose can be used to make ATP, but again, it can be converted to fat or to glycogen or even amino acids. So in priming, the second step of glycolysis, you have this isomerization of glucose 6-phosphate into fructose 6-phosphate, and then you phosphorylate again this fructose 6-phosphate to make it 1,6-diphosphate, and then this is going to be called priming because it will provide an activation energy, and this will help for the glycolysis cycle to proceed, and then you will have 2 ATPs consumed. Now in cleavage, the third step, you will have this cleavage of fructose 1,6-diphosphate into phosphoglyceraldehyde, which are going to be 3 carbon molecules. And then they are going to have, of course, since their name tells you phosphoglyceraldehyde, they are going to have a phosphate group attached to them. And then this phosphoglyceraldehyde is going to be oxidized when you remove these hydrogen ions, and then you put them into into NAD, so you can make NADH and hydrogen ions. And then you will have this phosphate group added into this phosphoglyceraldehyde, and then you will desphosphorylated it later so you can start forming pyruvate. So out of the 4 ATPs that you produce in glycolysis, which I remind you again happens in the cytoplasm, you have spent 2, but since you have generated 4, you have a net gain of 2 ATPs. And again, this is very little number of ATP to perform all the metabolic functions of the cell, so you have to put these pyruvates into the mitochondria so you can generate more ATPs. So these are the end products of glycolysis. So 2 pyruvates, 2 NADH, 2 ATPs, and 2 hydrogen ions. Now pyruvate, it can go into different pathways depending on the availability of oxygen. So if you don't have oxygen or mitochondria, like in the case of red blood cells, so the ATP is only generated through glycolysis. So glycolysis cannot continue, and then also you don't have glycolysis going on without a supply of NAD, and then this is going to be called what we know as anaerobic fermentation, and NADH is going to start making these electrons to flow into pyruvate so you can convert it into lactate, so you can regenerate NAD and carry more hydrogen ions into lactate, and this is anaerobic fermentation. Now lactate will go out of the cells, and then it will go to the liver, and then from there this lactate is going to be oxidated back into pyruvate by the liver, and we will be able to have some energy out of this. Now the liver is going to convert this lactate back into glucose 6-phosphate, and then it's going to start making glycogen out of this. And as it's doing this, it's going to remove the phosphate group and it's going to release it as free glucose. So we can have this glucose back into the blood so we can gain energy. Now, some of the drawbacks of anaerobic fermentation is that you undergo more steps, so you use more energy to generate glucose. And lactate itself, when you accumulate it so much, is toxic. Now, in a skeletal muscle, you have certain tolerance to these steps of anaerobic fermentation, and the cardiac muscle as well. But the brain does not employ anaerobic fermentation, so the brain always have to have oxygen. Fortunately, most of our ATP is generated through aerobic respiration, in which you always have oxygen available, and then this pyruvate will go into the mitochondria, and then it will enter into the Krebs cycle, which is located within the matrix of the mitochondria. And then through this Krebs cycle, you generate this NADH and then this FADH, so you can transfer these electrons into the electron transport chain. And then you can generate your ATPs. So the obtaining of ATP happens again within the cytoplasm, very little 2 ATPs, or within the mitochondria in aerobic respiration. So there are 3 steps to prepare pyruvate to enter into the Krebs cycle or citric acid cycle. So we have to decarboxylate this pyruvate, in which you remove CO2 and make it a compound that has 2 carbons. And then into these 2 carbons, you can convert them to an acetyl group or acetic acid, in which you can remove NAD plus and hydrogen from this C2 compound, and then you can bind these acetyl groups to coenzyme A. And coenzyme A is coming from this step from fat catabolism. So this is just to show you how this pyruvate can enter into the Krebs cycle, which is here. So you have this pyruvate in here, you decarboxylate it, so you form a 2 carbon compound, and then you remove hydrogen using NAD, so you form NADH, and then you end up with an acetyl group. To this acetyl group, you add coenzyme A coming from fat catabolism, so you can make acetyl-CoA. And then this acetyl-CoA now is going to be free to enter into this Krebs cycle, and then you join it with the last compound of this Krebs cycle, which is called oxaloacetic acid. And then when you mix this acetyl-CoA, or you combine this acetyl-CoA with oxaloacetic acid or oxaloacetate, you form citric acid, which is the starting point of the Krebs cycle. And then as you go into this Krebs cycle, you start taking away hydrogen from all of these compounds. Like in this case, you remove hydrogen from this part of the Krebs cycle and you form NADH, and again in here you form NADH, and you can also form FADH. When you do this, you have electrons that can be carried into the electron transport chain, which happens within the inner membrane of the mitochondria. And these are going to be important electrons because these electrons are going to be carried along these compounds or proteins within this electron transport chain that are going to be the responsible for generating the ATP. However, there is this part of the Krebs cycle in which you form ATP out of an analog molecule that you call GTP, or guanosine 3-phosphate. So you have GDP here, you phosphorylate this guanine diphosphate, and then you form GTP, and then this GTP you are going to make it into ATP. So acetyl-CoA, once you form it out of this pyruvate, you will combine it with oxaloacetate, or a compound that has 4 carbons, to form citric acid cycle, citric acid to form the start of the cycle. And then you remove water from this citric acid, so you will dehydrate it, and then you will remove these hydrogen ions or atoms, and then you will put this hydrogen into NAD, so you will reduce this NAD to form NADH, and then you can bring these electrons into the electron transport chain. So I already mentioned this, so let's move. So this is the final result of this citric acid cycle. So when you have 2 pyruvates coming from the glycolysis, and then you add 6 waters, and then you enter this into the citric acid cycle, you end up with 6 carbon molecules, 2 ADPs, 2 inorganic phosphates, 2 ATPs, 8 NADH, 8 hydrogen ions, and 8 NADH, 8 hydrogen ions, 2 FADs, 2 H, and then 2 FADH. So here is why our cells generate CO2 then. As you are forming ATPs, you form carbon dioxide. And then these will go out of the cells, enter into the circulatory system, travels into the lungs, and then you exhale them. And then as you're doing all these metabolic reactions, you will also generate heat. So the reactions then within this membrane in the mitochondria has 2 purposes. Is to oxidize NADH and FADH to transfer these electrons to form ATP, and to regenerate NAD and FAD so you can carry more electrons, and then you can form a link between the Krebs cycle and the electron transport chain. Now, within this membrane, we have a different set of compounds. We have one that is called the FMN, or flavin mononucleotide, that it is coming from riboflavin. And this is a compound that, as with the other ones, is bound to the membrane protein, and it will accept the electrons coming from the NADH. The next compound that we have in the membrane of the mitochondria, the inner membrane, is called iron sulfur, or FeS. And this will also be bound into the membrane as the other one, FMN. And this one doesn't accept the electrons from NADH, but it will transfer the electrons from FMN into the next compound, which is called coenzyme Q or CoQ. So CoQ will accept electrons from FADH, and this will move around the membrane to transfer the electrons into the next compound. Now we have also within the membrane copper ions, and we also have this complex that is called cytochromes. In cytochromes we have these subunits that are called beta, C1, C, alpha, and alpha 3. And then as these hydrogen ions are brought from the Krebs cycle into the inner membrane of the mitochondria, you will have the hydrogen ions splitting apart and releasing their electrons. And as they release their electrons, these hydrogen ions will become positively charged, so they will become protons, and they will be pumped into the intermembrane space. So the mitochondria has this membrane that it is formed out of an inner membrane and an outer membrane. And the space between the inner and the outer membrane is called the inner membrane. So you will transfer these hydrogen ions from the matrix into the intermembrane space as they release their electrons to be transported along the chain. And then at the end of these proteins that are embedded within the inner membrane, you will have the final acceptor of electrons, which is going to be oxygen. So oxygen will receive these 2 electrons coming from cytochrome A3, and then you will have 2 protons from the mitochondrial matrix forming water. So this is the diagram to show you how these complex are arranged within the inner membrane. So you have this FMN that is going to receive these electrons from NADH coming from the Krebs cycle. Then this FMN is going to be linked to this iron sulfur complex, which is going to be transferring some of these electrons into coenzyme Q. And this coenzyme Q is going to receive also electrons from FAD, FADH. And then if you see here in these two compounds, as this NADH and this FADH bring their electrons, they become oxidized. So they come in the reduced form, and then they release their electrons. They become NAD and FAD, so they become oxidized. And then CoQ is going to migrate between this complex and this other one that it is made by the cytochromes, and then you will transfer the electrons from cytochrome B into this iron sulfur complex, and then into cytochrome 1, and then into cytochrome C, and then into copper, and then into cytochrome alpha, and then into alpha 3. And then lastly, at the end of the chain, you have oxygen that is going to accept these electrons and is going to form water. So as this process happens, again, you will pump these protons in the inner membrane space. And as you're doing that, you are creating a gradient. So you will start accumulating little by little this hydrogen in the intermembrane space, and as it's getting more and more the gradient, these hydrogens will go back into the matrix through a pore in a protein in the inner membrane that is called the ATPase synthase. And this ATPase synthase is going to have ADP and inorganic phosphate, and it's going to bring them together. It's going to form a link between ADP and inorganic phosphate, so you will form ATP. So this is how then this transport of hydrogens happen. So here we have the matrix, this part in here. And then in here we have the inner mitochondrial membrane in orange. And then in here we have the outer mitochondrial membrane in red, which is this. This space between the inner mitochondrial membrane and the outer is called the intermembrane space. And as you are transferring these electrons between these complexes in the electron transport chain or electron transport system, you will pump these hydrogen ions out, as you can see here in complex 1, 2, and 3. As you are putting these hydrogen ions will form a gradient, so they will become higher in concentration, and then they will go back into the matrix. And they will pass through this pore in this channel, which is the ATPase synthase, and you will have ADP, and then you will couple this ADP with inorganic phosphate, so you will generate ATP. So for NADH, then every time that you transport these electrons into the electron transport chain, you will make 2.5 molecules of ATP, and when you put this FADH into this electron transport chain with its electrons, you will generate 1.5 ATP. So you will have there a total of 4 ATPs there. And then since you transport more NADH and FADH than just only one, when you are doing one turn of this cycle, of the Krebs cycle, you will generate a total of 32 ATPs. And then the rest of the molecules or energy that is coming out of the cycle is basically lost as heat. So pyruvate enters into the Krebs cycle, and then you generate out of this Krebs cycle 6 NADHs, and then you have 2 NADH coming from glycolysis and also from the transfer of pyruvate into the citric acid cycle. So you have 10 NADHs in here, and then you make here 2 FADH. So out of these NADH, these 10 NADH, if you remember, each of them when they transfer their electrons into the electron transport chain makes 2.5 ATPs, so you have 25 ATPs. And then each of these FADHs is going to give 1.5 ATP. So if you have 2 here, 2 times 1.5 will be 3. And then out of this GTP coming from the citric acid cycle, you have 2 ATPs. So if you add all of this in here, plus the 2 ATPs that you have coming from the net gain of the glycolysis, you end up with 32 ATPs generated per glucose molecule. So one thing about ATP is that we use it as its form, okay? And this is going to be an important molecule to transfer energy. It's not going to be used for storage. And our body stores energy as glycogen or fat. And then glycogen is going to be converted into glucose and fat as well, or amino acids, just in case that you run out of glycogen and fat, and then you start making glucose. Now, the reason for our body to use glucose is because at some point, yeah, there's a lot of steps that you have to undergo in order to form ATP. But this molecule of glucose is very simple. It's a 6-carbon molecule that you can break it easily and strip off the hydrogens to make ATP. Now, when it comes to proteins and fats, these are more chemically complex molecules. So our body tends to prefer glucose as a main source of energy rather than fats or proteins. So at some point, we first use glucose. Then if we run out of glucose, our body starts using fats. And then if we run out of glucose and fats, we start using amino acids or proteins. So then the formation of glycogen is known as glycogenesis. And glycogenesis is stimulated by the action of insulin. So remember, insulin, it is released by the pancreas whenever we have just eaten. And then this glucose is going to enter into the cells, is going to start undergoing this metabolism through glycolysis, and whatever is left is going to be used by our body to form storage, which is called glycogen, in muscle cells or in the liver. Now, if someone is fasting, this glycogen is going to be broken down into what we call glycogenolysis or hydrolysis of glycogen. So glycogenolysis is important because it helps to release glucose between meals, and the hormone that stimulates glycogenolysis is glucagon and epinephrine. So this is the antagonistic hormones for insulin. While insulin forms glycogen, glucagon and epinephrine breaks down glycogen. And then the glycogen that we use to maintain the glucose levels that are necessary to be in the blood is going to be coming from the liver cells. So we use the glycogen from the liver cells. Muscle cells, they don't give their glycogen. They use it for their own contraction. And why do we have to have glucose in our blood? Well, because the brain uses glucose as its source of energy. Okay, so we have to have glucose for our neurons to be happy. Now, since glucose is very important, whenever we form glucose out of substrates that are not carbohydrates like fat or proteins, this is called gluconeogenesis. And gluconeogenesis happens within the liver, but it can also happen in the kidneys. So these are the major pathways for storage and use of glucose. So glycolysis, it is used to generate ATP, so to use glucose. Okay, and we can have excess of glucose, and then we can form glycogen through glycogenesis. And this glycogen can be broken down whenever we have not ingested food into glucose 1-phosphate, which it can be used in glycolysis into what we call glycogenolysis. Now for lipid metabolism. So triglycerides will be the stored form of lipids in our body, and our lipids are not static. We have this changing or transformation of lipids every 2 to 3 weeks. And these lipids are released into the bloodstream, and then they can be either oxidized or deposited into other fat cells. And then whenever we are making fat from other types of molecules that are not the ones that we ingest, it's called lipogenesis. So amino acids and sugars can be converted into fatty acids and glycerol, so you can form triglycerides. And phosphoglyceraldehyde out of the glycolytic cycle can be converted into glycerol, and acetyl-CoA can be used to make fatty acids. So pyruvate can be converted into acetyl-CoA, enter into the Krebs cycle to generate ATP, or it can be used to make fatty acids. And this phosphoglyceraldehyde, if you remember, this is the glycolytic cycle in here. So this phosphoglyceraldehyde can be converted into glycerol. So now acetyl-CoA comes from the breaking down of lipids, as I told you before. So if you have stored triglycerides in the adipocytes, the cells that store the fat, you can form glyceraldehyde. Sorry, you can form glycerol, which is going to be converted into phosphoglyceraldehyde. So you can generate pyruvate, so you can have energy through the citric acid cycle. But this glycerol and this phosphoglyceraldehyde can be going into make new triglycerides when you combine them with fatty acids, and you can have a recycling point in here. Now, fatty acids can undergo what we call beta oxidation and then form acetyl-CoA. And then it can also form acetyl groups rather than acetyl-CoA, and into what we call ketone bodies, which are beta hydroxybutyric acid, acetoacetic acid, and acetone. So before we move into this, just remember ketone bodies are toxic, but they can be used by our brain just as a desperate measure when someone doesn't have energy coming from glucose. So lipolysis is the breaking down of fat for fuel, and it begins with the hydrolysis of triglycerides into glycerol and fatty acids. Lipolysis is stimulated by several hormones, including epinephrine, glucocorticoids, thyroid hormone, and growth hormone. So when someone is under stress, generally it will release epinephrine and then glucocorticoids, and then you start mobilizing all this fat storage, and then you can have energy to face that stressful situation. Now, one glycerol molecule, it can generate only half as much ATP as glucose. Beta oxidation of lipids happens within the mitochondrial matrix through removal of 2 carbons at a time out of this long chain of carbon molecules that we have in the fat. And then you will form acetyl-CoA that it can enter into the citric acid cycle. Now, fatty acids with 16 carbons can yield up to 129 molecules of ATP. So this is a much rich source of energy than the glucose molecule. So remember, glycerol doesn't form so many ATPs, but fatty acids coming from triglycerides, they form up to 129 molecules of ATP. So it's a very good molecule. And then ketogenesis, the formation of ketone bodies, happens within the liver. And I already mentioned the names of the ketone bodies. Again, these are toxic to the body, but our cells can use them as a desperate measure, especially the brain cells. So, ketosis is the rapid or incomplete oxidation of fat, and this can lead to decrease in the pH into what we call ketoacidosis. Now, the amino acid pool is important for protein metabolism. So, whenever we have dietary amino acids plus 100 grams of tissue protein broken down each day into free amino acids, you can use them to synthesize new proteins, and this will be the fastest rate of protein turnover in the intestinal cells, and this will be frequently replaced. Now, out of all the amino acids that are absorbed by the small intestines, 50 will come from the protein diet, 25% will be coming from the epithelial cells that die, so we recycle them, and 25% will come from enzymes that has digested each other. So our body at some point doesn't waste the proteins also coming from the enzymes, so we recycle them. And then these amino acids that are coming from whether the absorption through the nutrients, the recycling of the dead epithelial cells, or the enzymes, we will use them to form this pool of amino acids that we call sometimes free amino acids, so we can make glucose and fat that can be used as fuel. Now, we have this exchange of amino acids into what we call transamination. And we can have amino groups added into these molecules into what we call amination, or we can remove these amino groups into what we call deamination. Now, if you want to use amino acids as fuel, you have to deaminate them or remove the amino group. So after you remove these amino groups, you have something that is called a keto acid. And this keto acid can have several fates. It can go into make pyruvate, it can go to make acetyl-CoA, or it can be converted into one of the acids that enters into the citric acid cycle. And then these reactions are reversible if you have deficiency of certain amino acids. And these keto acids that you form through deamination can enter into gluconeogenesis, so you can form glucose. Now, when you deaminate an amino acid, you will have these molecules like intermediates that helps to make other type of amino acids what are more necessary. For instance, you can have alpha ketoglutarate acid converting into glutamic acid, and then glutamic acid can go into the liver, and then you can form again alpha ketoglutarate acid, and then you can form this ammonia, which is toxic, but remember the ammonia can be eliminated through the urea cycle. As you enter this ammonia into the urea cycle, you form urea, and then urea is going to be excreted by the kidneys. Now also you can synthesize proteins when you have this excess of amino acids, and you can use these amino acids to make proteins that are involved in DNA and mRNA, messenger RNA, transfer RNA, and ribosome synthesis. And this synthesis of proteins, it is stimulated when you have these amino acids available in your diet and you have growth hormone, thyroid hormone, or insulin. Insulin in general is an anabolic hormone. And in order to form certain proteins, you have to have, hopefully, all the amino acids. We have 20 amino acids, and then some of them are called essential because they must be obtained from the diet because our body cannot transform these or form these amino acids from others. And then we have non-essentials that are called non-essential because they can be made by our body, especially by the liver. So these are then the pathways of amino acid metabolism. So in this case, you have the glycolytic cycle, how it connects to the citric acid cycle. But out of the citric acid cycle, you can have alpha ketogluteric acid that is going to be combined with this amino group. So you aminate it to form glutamic acid. And then you can have a cycle in here, and then the ammonia that is generated is going to enter into the urea cycle, and then you form urea, which is going to be eliminated by urine. Now the keto acids can go, after you deaminate these amino acids coming from the protein pool, you can enter them into the citric acid cycle through acetyl-CoA, or through the formation of these amino acids or keto acid that can enter directly into the citric acid cycle, like ketoglutaric acid, or they can go and form glucose into what we call gluconeogenesis. And when you have excess of amino acids, you can form proteins. Now, our metabolism changes frequently, depending on how long had you had your reserves available since you have eaten your last meal. And then we have this part of the metabolism that help us to absorb the nutrients that we call the absorptive or fed state. And this will last 4 hours during and after the meals. And in this case, your nutrients will be absorbed, and then your nutrients will be used immediately to meet energy needs. And after you have eaten your meals, you will enter and you absorb them, you will enter into what is called post-absorptive or fasting state, which will be this state that prevails in the late morning, late afternoon, and overnight. In this case, you don't have nutrients available, and then you will have this empty stomach or intestines, and your body energy will meet from the triglycerides or the glycogen, hopefully first. But if you don't have glycogen and if you don't have triglycerides available, you will start using your proteins. And this is bad because proteins are a very important part of our tissues. Okay, so before we move into the next slide, then let's consider our key points. So when NADH is reduced, it can be converted into NAD. So it will be oxidized. And when NAD is reduced, it will convert into NADH. We will consider this in just a few slides, but vitamins A, D, E, and K are water soluble, and vitamins B and C group are, sorry, it's the other way. Vitamins A, D, E, and K are lipid soluble, while vitamins B and C are water soluble. Uh, Now, most of the ATP from metabolism is produced in the electron transport system or electron transport chain. And in oxidative phosphorylation process, the energy for synthesis of ATP is directly coming from the flow of hydrogen ions through the ATPase synthase within the inner mitochondrial membrane. What are the end products of glycolysis? ATP, NADH, and pyruvate or pyruvic acid. During lipolysis or breaking down of fats, triglycerides are broken down into glycerol and fatty acids. HDL is called good cholesterol. HDL or good cholesterol is called good cholesterol because it carries excess of cholesterol from peripheral tissues to the liver. And again, to reemphasize, when NADH is reduced, it converts into NADH. When NADH is oxidized, it converts into NAD. Okay, so in the absorptive state, glucose is going to be available for ATP synthesis. And then glucose is going to be the primary fuel that is going to be used by our tissue cells, and whatever is left is going to be stored as glycogen or fat. Now the carbohydrates are going to be absorbed by the bloodstream. So you will have the hepatic portal vein that is going to transport these carbohydrates into the liver so that the liver can metabolize this glucose or these carbohydrates. And then your liver is going to convert the excess of glucose into glycogen or fat, and most of the fat synthesized by the liver is going to be released into the circulation. Now the fats, once they are into the circulatory system, are going to be stored by the adipocytes as triglycerides. So we have this enzyme in the intestinal cells that are called lipases that helps to break down these triglycerides so we can absorb them. And in the enterocytes or the cells of the intestines, we have these lacteals that will absorb these fats, and they will form what we call the chylomicrons. And then you will have these chylomicrons traveling through the lymph, and then you will enter these chylomicrons into the circulatory system. So you will have this fat coming now into the liver, and then the liver can dispose of the chylomicron remnants, and then your fats will be used as primary energy substrates by first the liver and then the fat cells and the muscle cells. Now, what is going to happen with the amino acids is that they will pass through the liver and then they can go into the other cells for protein synthesis. In the liver, these amino acids can also be used for protein synthesis, but they also can be used for ATP synthesis or fatty acid synthesis. So what regulates the absorption of our nutrients or our glucose will be insulin. And also you will have intestinal hormones like gastrin, secretin, cholecystokinin, and glucose-dependent insulinotropic peptide or GIP. Now, insulin specifically is going to help to absorb this glucose because it's going to help to activate these glucose transporters so that you can have this absorption of glucose into the cells. So there are so many effects of glucose on target cells. So first, it's going to increase the cellular uptake of glucose, so you will have a decrease of glucose in your bloodstream, and then your glucose is going to be oxidized in glycolysis, just in case that you need this glucose to be transformed into ATP. But also, if you have excess of glucose, you will form glycogen in glycogenesis, or you can form storage of fat into what we call lipogenesis. So insulin is going to inhibit the formation of glucose out of amino acids and proteins. So it's going to inhibit gluconeogenesis. And it's going to also stimulate the transport of amino acids if you have amino acids in your bloodstream, so you can form proteins. So again, insulin, it's an anabolic hormone, and is going to send a signal into the brain that acts as an adiposity signal. So it will send a signal to tell our brain the index of fat stores. And when you have high amino acids, you will stimulate the release of insulin and glucagon, supporting adequate levels of glucose to meet the needs of the brain. In the post-absorptive state, you will have a glucose concentration between 90 to 100 milligrams per deciliter, and this is important because your brain uses a lot of glucose, so you have to have at minimum 90 milligrams of glucose per deciliter. And then you will use, just in case that you run out of glucose, you will start using glycogen first, then you will start using fats, and then you will start using proteins, just to maintain those levels so that your brain cells can have enough glucose available for energy. So what is the status of the major nutrients during post-absorptive state? So your glucose is going to be drawn from glycogen reserves or synthesized from fats or amino acids into what we call gluconeogenesis. Now, the major organ that is going to release glycogen is going to be the liver, and that glycogen is going to provide glucose for around 4 hours. If 4 hours passes by and then you don't ingest food, your fat cells, your adipocytes, are going to make glucose. So you will start hydrolyzing these triglycerides, so you start releasing glycerol-free fatty acids into the circulation, and then you start gluconeogenesis. And at the same time, you start also forming ketone bodies. And then proteins will be the last reserve for making glucose. And out of the proteins that we have in our body, usually collagen, it is preserved, so it's almost never broken down for fuel, but actin and myosin in the muscle will start releasing their amino acids so you can form proteins. So you can reach up to a point where you lose so much protein that you call this cachexia, in which you have extreme wasting away of proteins. But in this case, usually it's when you have diseases like cancer or any other chronic disease. The hormones that regulate, or the hormone that regulates this post-absorptive state, will be glucagon, the antagonistic hormone of insulin. So this glucagon is going to promote glycogenolysis and gluconeogenesis to raise blood glucose levels. And it will promote lipolysis to rise the levels of free fatty acids and makes both glucose and lipid available for fuel. Another thing that is going to happen during post-absorptive state is that your adrenergic system or sympathetic system is going to promote glycogenolysis and lipolysis. During stress time, you release cortisol. I already told you this before, and this is going to mobilize fat storage, and it's going to start also catabolizing proteins, so you can have glucose available. Now, the metabolic rate is the amount of energy free up or liberated in the body in any given time. It is measured in kilocalories per hour or per day. And this usually is measured with a device that we call calorimeter, in which you have a closed chamber with water-filled walls that will absorb heat given off by the body. And this is the most used apparatus. We have another one that is called a spirometer. That is not the one that we use in the lab for measuring the PFTs or pulmonary function test, but it's different. It's one that tells us how much oxygen a person consumes. Now, our metabolic rate will depend on several variables, including physical activity, your hormonal state, your absorptive post-absorptive status, your age, genetics, levels of thyroid hormone, etc. BMR, or basal metabolic rate, will be a baseline or a standard that compares the minimal effects of activity, feeding, and hormonal levels. So an adult BMR is usually in the males 2000 kilocalories per day, and it's slightly less for females. Your total metabolic rate, or TMR, will be the sum of BMR and the energy expenditures for all your voluntary activities. Some factors that increases the TMR are physical activity, pregnancy, anxiety, fever, eating, your levels of epinephrine or epinephrine, thyroid hormones, and TMR is high in children because children are growing their tissues, their muscles are growing, everything that the tissues in the body in children are growing, so they have a greater TMR than adults. And then some factors that can decrease the TMR will be depression, prolonged starvation, inactivity, hormone levels as well. If you have low levels of thyroid hormone, you will start gaining weight, etc. Now, our appetite, it is controlled by different hormones. So we have something that we call the gut-brain peptides that sends a signal to the brain, and some of these act as short-term regulators of appetite, which makes one feel hungry and then you start eating, or make one feel satisfied and you stop eating. And these are regulators of appetite that works over a period of minute 2 hours. So what are some of these hormones? Will be ghrelin, peptide YY, and cholecystokinin. Ghrelin help us to feel hungry or hunger, and then this will stimulate the hypothalamus, so we start eating, and then once you feel satisfied, you stop the secretion of ghrelin. Peptide YY, this is secreted by cells that we have in the ileum and the colon that we call enteroendocrine cells, and then they will sense when our cells in the stomach has received these chemicals that are coming from food. So the PYY, it is secreted long before the chyme reaches the ileum. So the first sensors for the sending of the signal starts before reaching the ileum. And then we secrete PYY according to the calories that we have consumed. If you have eaten a big meal, you secrete more PYY. And this PYY, it will have an effect in the brain, so they will send a signal of satiety so we can stop eating. Can this be mutated in some persons? Yes, it can be. That's why many people even have eaten a large meal and they can continue eating. Cholecystokinin, or CCK, it is secreted by cells in the duodenum and in the jejunum that are also enteroendocrine. They will, once you release cholecystokinin, you have contraction of the gallbladder, so you secrete bile and pancreatic enzymes, but also they will stimulate the brain and the vagus nerve so you can suppress appetite. So PYY and CKK will send a signal into the hypothalamus in our brain so we can stop eating. Another hormone that is a short regulator of appetite is amylin, that it is secreted by the beta cells in the islet of the pancreas that helps to produce satiety sensation and inhibits the stomach from moving. Now we also have long-term regulators of appetite that works over a period of years, weeks up to years, like leptin and insulin. So leptin, this is a hormone that is secreted by the adipocytes, and it will tell the brain how much body fat do we have, and actually helps to suppress appetite. In general, we secrete this hormone while we are eating. So sorry, when we are sleeping. So in order for us to kind of control our appetite, we have to also sleep well. So when someone is deprived of sleep, the person will start secreting less leptin, and then the person start feeling less satisfied, and the person will eat more, and consequently will gain more weight. So it is important as well also to sleep well for controlling our body weight. Now insulin is secreted by the beta cells in the pancreas, will stimulate the intake or uptake of glucose and amino acid by our cells, so it will help us to form glycogen and also triglycerides, so we can have fat storage. And then it will similarly work as leptin, so it will provide an index of to the brain, an index of body fat stores, and it will have a weaker effect on suppressing the appetite than what leptin do. Specifically, the center in the brain that we have that help us to control appetite is called the arcuate nucleus, that is located in the hypothalamus. And then you will have a couple of neural networks that are involving in hunger. So one of the groups of the hypothalamus of this arcuate nucleus will secrete this neuropeptide Y that is going to stimulate the appetite, and we have the other group that is called melanocortin that you inhibit eating, while the group that secretes the neuropeptide Y, it will work with ghrelin to stimulate the neuropeptide Y secretion, and also with insulin, PYY, and leptin to inhibit it. The other group is going to work with molecules that helps to inhibit eating, and this melanocortin group is going to be inhibited by appetite stimulants like endocannabinoids. So here is the diagram of the regulators of appetite. So here we have these MPY secrete neurons in the hypothalamus, in the arcuate nucleus, and then we have here these melanocortin secreting neurons. So they have opposite effects. This one, melanocortin, is going to send a signal into the cerebral cortex so we can feel satisfied, and this other one, the MPY, is going to activate the cerebral cortex so we can feel hungry. And leptin is going to have an stimulatory effect on melanocortin, and insulin is going to inhibit together with CCK and PYY this MPY so we can feel satisfied. And ghrelin is going to activate this MPY so we can feel hungry. Now, hunger is stimulated also by peristalsis. So when we have mild hunger, we'll start having this peristalsis that begins soon after the stomach is empty, and then this will increase in intensity of a period of hours, and it doesn't affect the amount of food that it is consumed. Now, appetite is briefly satisfied by chewing and swallowing, but also is going to be briefly satisfied as the stomach is getting filled, and it will have lasting satiety depending on what type of nutrients enter into the blood. So at some point, out of the 3 nutrients that we can ingest, proteins have a better effect on decreasing our appetite than glucose and fats. Glucose is used right away by our body. So the glucose levels are going to drop really easily, but amino acids, since they're more complex in their molecule, they will take longer to be metabolized. So that's why you feel more satisfied after you have eaten a protein-rich meal. And as well, depending on the quality of the nutrients. So now the neurotransmitters that stimulate the desire for a different type of food will be norepinephrine. When you have norepinephrine in your bloodstream, you will have cravings for carbohydrates. When you have galanin, you will have craving for fats, and when you have endorphins, you will have cravings for proteins. Now, excess of body weight greater than 20% is obesity, and in US we have 30% of obese people, and another type of consequence is having overweight, which is 35% of the population. Now, the BMI, or body mass index, is an indication of overweight or being obese. Body mass index is calculated by dividing the weight over the square height in meters. And a normal body mass index is between 20 to 25. And if someone has over 27 but below 30 is considered to be overweight, and if someone has above 30 is obese. Now we have other indexes for extremes of obesity, but greater than 30 BMI is obese. Now, some problems of obesity is that decreases the life expectancy, because obesity can pose a risk for life, because someone can accumulate plaques of fat inside their blood vessels, in what we call atherosclerosis. The person will have great chances of having hypertension, diabetes, etc. Causes for obesity are diverse, not because someone overeats. It can mean that the person is going to become obese. Sometimes it's just hereditary. You don't eat that much, but you have genes for storing this food into fat rather than utilizing it. Or you can have also problems in the endocrine system like hypothyroidism. Now, we measure the amount of energy in calories. 1 calorie is the amount of heat that is required to raise the temperature of 1 gram of water to by 1 degree Celsius. And 1000 calories with lowercase is equivalent to 1 kilocalorie, or what we write down as 1 calorie with capital C. And this is a measure of the capacity to do biological work. So carbohydrates and proteins, they yield 4 kilocalories or 4 calories per gram. And fats, they yield 9 kilocalories per gram. And alcohol yields 7.1 kilocalories per gram, but they are empty calories. So in order for us to have good nutrition, we have to have a balance of the amounts of protein, lipids, vitamins, and other nutrients that meet our body requirements. Body requirements vary according to your age, your gender, your physical activity, your physiological state. And we use then these nutrients coming from all of these, or we use the ATP or the structure out of these nutrients to meet our needs in order to make ATP. So a nutrient is defined as any ingested chemical that it is used for growth, repair, and maintenance of the body. Nutrients are classified as water, carbohydrates, lipids and proteins, vitamins and minerals. Water, carbohydrates, lipids, and proteins are macronutrients because we have to need consumption of them in large quantities, while vitamins and minerals are considered micronutrients because we don't require a lot of amounts of these, but they still are necessary. We consume different types of nutrients in different types of amounts based on DRIs or dietary reference intakes, and these are recommended by the FDA in what we call recommended daily allowances or RDAs. These RDAs are the safe estimates of daily intakes that will meet the nutritional needs of most healthy people. And then adequate intake is a less certain established parameter for the different types of nutrients. Now, in general, we have essential nutrients that cannot be synthesized by the body, like minerals, vitamins, or most of the vitamins, and 8 amino acids, or 9 if they are children. And then 3 fatty acids are required by our diet. These are called essential. So carbohydrates, we supposed to have around 440 grams in a well-nourished adult. And out of these carbohydrates, we're supposed to have 325 as glycogen in the muscle, 90 to 100 grams in the liver as glycogen, and 15 to 20 grams in the blood. Functions of the carbohydrates will form glycoproteins, glycolipids, and to start forming also nucleic acids and ATP. And they can serve as fuel if they're used directly through glycolysis and the citric acid cycle. And the neurons and the erythrocytes depend solely on carbohydrates. And hypoglycemia will be deficiency of blood glucose that can lead to dizziness and then fainting because your nervous system depends on glucose. Glucose levels are regulated by insulin and glucagon. While insulin decreases glucose levels, glucagon increases it. And again, excess of carbohydrates are converted into fat for storage and also into glycogen. The RDA for carbohydrates will be 130 grams, and this is because the brain alone consumes 120 grams of glucose per day. The major forms of carbohydrates that we ingest are monosaccharides like glucose, galactose, and fructose, or disaccharides like sucrose, which is a combination of glucose and fructose, or maltose and lactose. And then we can also consume polysaccharides like starch, or cellulose, which is not a nutrient because it's fiber. So that's why we don't use it for energy, just to make a bulk. And then glycemic index will be a measurement of how much blood glucose levels can be influenced for the intake of certain foods. So some dietary carbohydrates can increase the glucose levels much faster than others, so they will have a high glycemic index. And whenever you have food that has high glycemic index, you will stimulate the release of insulin much faster, and that will have a greater risk to have proneness for developing obesity and type 2 diabetes. Now, dietary fiber will be all the fibrous material of the plant and an animal that resists digestion. Examples will be cellulose, pectin, gums, and lignins. The RDA for fiber will be 25 grams in females and 38 grams in males. Now, we have water-soluble fiber like pectin that is found in foods like oats, beans, peas, brown rice, and fruits that helps to decrease blood cholesterol levels and LDL levels, which is low-density lipoproteins or bad cholesterol. Water-insoluble fiber like cellulose, hemicellulose, and lignin don't have an effect on cholesterol and LDL, but helps to absorb water in the intestines to soften the stool and increase the bulk, stretching the colon and stimulating peristalsis so you can have better digestion. Lipids in the body of males form 15% of storage, while in females we have greater content, 25% of our body weight will be fat. And a well-nourished adult will meet 80 to 90% of the resting energy needs from fats. So fat is superior to carbohydrates to form an energy storage for two reasons. Carbohydrates are hydrophilic, so they will absorb a lot of water, will be so bulky, so it will occupy more space, while fats, they are hydrophobic, so they are more compact in a structure, and they will provide over twice as much as the energy of glucose or carbohydrates. So 9 kilocalograms for fat as opposed to 4 kilocalograms of energy provided by the carbohydrates. Now, fat has a glucose sparing effect and protein sparing effect. So that's why fat is used as a storage form. Now we have fat-soluble vitamins like vitamins A, D, E, and K that are absorbed together with dietary fat. And ingestion of less than 20 grams of fat per day will risk vitamin deficiencies of A, D, E, and K. Functions of the lipids will be to form, or fats will be to form phospholipid within the plasma membrane of the cells, Also, fats like cholesterol helps to form rigid plasma membrane, not so rigid but more rigid than without it, and also helps to form myelin. And remember, cholesterol, it's an steroid, so it will help to form uh, sex hormones like estrogens, testosterone, and also bile salts that are important for emulsification of fats, and also help to form vitamin D and clothing factors like thromboplastin, and form chemical precursors like fatty acids that are going to be transformed into arachidonic acid, linoleic acid, that are going to form chemical messengers like prostaglandin and eicosanoids, and also as well help to cushion our organs and protect our organs. So they have protective and insulating functions. Requirements for fat will be 35% of calorie intake. Less than 10% of fat are saturated fats that we require. And we have a typical diet that has 40 to 50% fats. And out of these fats, we have these essential fatty acids that need to be consumed. Sources of fat would be meat, egg, yolks, dairy products, coconut, palm oil, nut seeds, etc. So, for transport of lipids. So, the lipids must be transported to all the cells of our body, and they are hydrophobic. So, they need to be transported with these lipoproteins. These lipoproteins, they form these complexes with the lipids, and they will be circulating throughout the body. So the serum lipoproteins are classified according to their density into chylomicrons, very low density lipoproteins, low density lipoproteins, and high density lipoproteins. So here are the different forms of lipids and their composition. So chylomicrons, they have 3% phospholipids, they have 90% triglycerides, 5% cholesterol, and 2% proteins. Very low density lipoproteins, they have very low protein, 8%. They have high amount of triglycerides, 55%, low amount of cholesterol, 20%, and 17% of phospholipids. Low density lipoproteins, or LDLs, they have low amount of triglycerides, 53% of cholesterol, and 20% of protein. So this LDL is bad cholesterol. This has a lot of cholesterol in it, and this can clog our arteries. Now the good cholesterol, it is the HDL. It has 50% of protein, 20% of cholesterol, 5% of triglycerides, and 25% of phospholipids. Again, the fats are transported first, or triglycerides, as chylomicrons, and they enter into the lymph first and then into the bloodstream. And as they are entering into the bloodstream, they do so by passing through the endothelial cells with the aid of this enzyme that is called lipoprotein lipase. So you can break down these triglycerides to form monoglycerides and free fatty acids, and then you can regenerate them inside the intestinal cells, and then you can form these chylomicrons that goes into the circulation, goes into the liver, and in there the liver can process them as VLDLs or LDLs. And then these VLDLs and LDLs will distribute these triglycerides into the different tissues, into adipose tissue, or into the cells so that the cells can use this as source of energy. Now the HDL is going to carry all the cholesterol from the peripheral organs and bring it into the liver so that the liver can recycle this cholesterol as bile or bile acids. So HDLs remove excess of cholesterol from the body, so you don't have clogged arteries. Now, we have this concentration of plasma cholesterol, which is supposed to be less than 200 milligrams per deciliter. If someone has between 230 239 cholesterol, is going to be borderline high cholesterol, and if someone has greater than 240 milligrams cholesterol in their bloodstream, this is going to be pathological because it can clog our arteries, so it can form plaques, and then you can have this myocardial infarction. Now, independently of your diet, either you might not opt to ingest cholesterol but our body needs to synthesize cholesterol. So it will synthesize cholesterol to make the cholesterol that goes into the phospholipids of the plasma membrane, or to make the different hormones that are coming from this cholesterol. It is known that exercise lowers cholesterol, so that's why it's one of these recommendations that is given to persons who have high cholesterol levels. So, recommendations to increase the ratio of HDL to LDL: regular aerobic exercise, stop smoking, and stop ingesting fats that are saturated, and stop drinking coffee, and the person being less stressed. So this is how you process these lipoproteins. So you can absorb these lipoproteins with your small intestine, then bring it into the circulatory system through this lipoprotein lipase, and then from there your triglycerides can go into the adipocytes for being stored as fat, or it can go into the cells so they can be used as source of energy. And some of these they will go into the liver, and then the liver will form VLDLs, and then these VLDLs can be transformed into LDLs, and then you can have these triglycerides going into the different tissues, and then your HDL will bring the excess of cholesterol into the liver so that the liver can recycle this cholesterol as bile or bile acids. Now for the proteins, they form 12 to 15% of our body mass. 65% of the proteins is found in the skeletal muscle, and they have several functions. Among some of them is contraction of muscles, motility of the cilia and flagella within the cells, forming protein channels, forming fibrous proteins like collagen fibers, collagen within cartilage, bone, within tendons, ligament, etc. Also, some proteins are forming a transport system like globular proteins. Some of them they are antibodies. Some of them they are carriers of drugs like albumins, etc. And they also contribute to the resting membrane potential of all cells because they form these sodium-potassium pumps. Recommended daily protein intake will be 46 to 56 grams per day. Again, this is depending on your age, body weight, whether you're female or male, your physical activity, And then you can calculate that by multiplying your weight in pounds times 0.37. So physiological states where you require more intake of proteins. Well, when someone is growing, children, if someone has stress, if a female is pregnant, etc. Now, when you over intake proteins, you can generate more nitrogenous waste that is going to be processed by the kidneys, but as the kidneys are processing this, you can have kidney damage. So out of these proteins, we break down these proteins to make amino acids, and then we have here the the 8 essential amino acids, and the 12 non-essential ones will be the rest. So we have 20 amino acids in total. There are 9 essential in children, but 8 in adults. And the cells do not store surplus of amino acids for later use. So when a protein is synthesized, all amino acids must be present at once. If one is missing, the protein cannot be made. In our diet, we can have complete and incomplete proteins. Complete proteins will be all the ones that has all the amino acids, and incomplete proteins will be the ones that do not have all the amino acids, including the essential ones. And then in order for us to compensate for that, we can have a diet buried in incomplete proteins, so you can make out of the incomplete proteins a complete protein. Our proteins can come 70 to 90% out of animal products, while 40 to 70% out of protein products, or plant products. So the advantages of having proteins in our meals is that you will have more vitamin, minerals, and fiber available because they help to decrease the excretion of them, and you can have also less saturated fat, no cholesterol, less pesticide. Now, animal proteins, they closely match our human proteins, and you can opt out if you are not someone who ingests animal proteins. You can have your proteins coming from plant sources that combined can give the right proportions of animal proteins. Now, proteins, they contribute to the formation of nitrogenous wastes, and in order to have homeostasis, you have to have a nitrogenous balance or nitrogen balance. So in this case, your rate of nitrogen ingested will be equal to the rate of nitrogen excreted, and you can have positive nitrogen balance in which you have more ingestion of nitrogen or more ingestion of proteins than whatever you increase, and this will occur in children because they retain more protein for tissue growth. Also, this can happen in pregnant women and athletes because they have this also requirement of retaining some proteins as they're making tissue. And you have growth hormone, sex steroids that promotes protein synthesis and positive nitrogen balance. Negative nitrogen balance will be when you have more excretion than ingestion. Example would be when you have poor diet in proteins, and you start breaking down your proteins. Okay, lastly, for minerals and vitamins. Minerals are inorganic elements that are extracted from the plants and also from the soil or water, and then they're introduced into food web. And then vitamins are organic compounds that are necessary for metabolism. Out of these vitamins, we have these cofactors that are needed for metabolism to happen. And vitamins and minerals are not used as fuel, so they are at some point essential for our metabolic pathway so we can use the other nutrients. Minerals form 4% of our body mass, so 3/4 begin being calcium and phosphorus in bones and teeth. And then phosphorus is found as a component of phospholipids, ATP, cyclic AMP, GTP, and creatine phosphate. So minerals other than calcium, we have iron, magnesium, manganese that are cofactors for enzymes. Other minerals that are important is iron for making hemoglobin, myoglobin, and chlorine, which is part of the hydrochloric acid in the stomach. And they have several functions, including helping the nerve cells and the muscle cells to function. And sources of minerals will be vegetables, legumes, milk, eggs, etc. Okay, for salt, we have 1.5 grams per day as the RDA, and we consume more than that. Whenever someone consumes more salt than what it's supposed to, the person can have chances of having hypertension. For vitamins, so we have this division into water soluble again and to lipid soluble. Water soluble will be vitamins B group and C, and for lipid soluble will be A, D, E, and K. Now niacin is part of the vitamin B group and is derived from tryptophan. Vitamin A is derived from keratin, which is a pigment that it is found in carrots and other vegetables. Vitamin D, it can be synthesized from cholesterol, and vitamin K can be synthesized by bacteria in the large intestine. Also, pantothenic acid, biotin, and folic acid can be produced also by bacteria in the large intestine. So water-soluble vitamins are absorbed with water in the small intestine and can be excreted by the kidneys. They are not stored. Another name for vitamin C, which is a water-soluble vitamin, is ascorbic acid. It's important to make hemoglobin, to make collagen fibers, and to make strong connective tissue. It is an antioxidant that helps us to protect our cells from free radicals of oxygen, and it helps to reduce the chances of having cancer because of that, because it's an antioxidant. Vitamin B group function as coenzymes for the electron transport chain, for any other metabolic pathways within the body. And fat-soluble vitamins, they are absorbed with micelles in the small intestine or small globules of fat. And vitamin A is important to make retinal pigments that are important for us to see and help also to promote the synthesis of connective tissue components like proteoglycans that helps to maintain epithelial cells of the skin and the mucosa. Vitamin D is important for the absorption of calcium and phosphate in the intestinal cells. Vitamin K is important for forming prothrombin, which is a clotting factor. Vitamins A and E are antioxidants, just like ascorbic acid, help us to protect from free radicals of oxygen, preventing cancer. So, excess of vitamins are known as hypervitaminosis. Depending on the type of vitamin that you are talking about is if you have hypervitaminosis. Hypervitaminosis rarely happens with water soluble because they are excreted by the kidneys, but it can happen with fat soluble, like with vitamin A. In this case, vitamin A can be stored in our body, and it can cause anorexia, nausea, vomiting, disturbance of the nervous system, that it can actually lead up to coma. Okay. Now, for deficiencies, it can happen in any kind, whether it's in water soluble or vitamin or lipid soluble. For instance, in vitamin A, if someone doesn't have enough vitamin A, the person will not form this pigment in the retina, and the person will have night blindness. It can have problems in the skin because it won't have a strong connective tissue, so the person will have dry skin, dry hair, etc. Okay, so that's all that we have for this chapter. If you have any questions, email me, contact me, guys. Don't leave your questions blank. Okay, so have a good day or have a good night. Again, this is all for this chapter. Bye bye.